Nested PCR. In nested PCR, two successive steps they are used by using two set of primers. कि इसमें हम दो PCR जो रिएक्शन है अ, वो यूज़ करते हैं और दो ही सेट ऑफ प्राइमर हम इसमें यूटिलाइज करते हैं. So actually what happens in nested PCR is that in first PCR reaction or mixture, the target DNA it is amplified by using one set of primer, and this PCR mixture will lead to amplification, and it will accumulate the DNA material in the sample, that is target DNA it is amplified by using first set of primer. Now the product that is generated from the first reaction, it is subsequently used or subjected to second PCR reaction. And for the second PCR, we used second set of primers to amplify the DNA molecule that is generated from the first PCR. कि second step में हम क्या करते हैं कि जो फर्स्ट रिएक्शन में हमारे पास डीएनए आया है, बाय यूजिंग फर्स्ट सेट ऑफ प्राइमर उस प्रोडक्ट को लेते हैं फ्रॉम द फर्स्ट रिएक्शन और सेकंड सेट ऑफ प्राइमर से लेके उसको फर्दर एम्पलीफाई करते हैं. और इसमें जो सेकंड प्राइमर है वो फर्स्ट प्राइमर वाली पोजीशन से अंदर अटैच करता है. एंड बिकॉज ऑफ दिस रीजन इट इज़ कॉल्ड एज नेस्टेड, एंड दिस टर्म इट इज़ derived फ्रॉम नेस्ट, that is to lie as inside to something, कि किसी चीज़ के इनवर्ड पाया जाना. कि जो फर्स्ट प्राइमर है उसके जो अंदर वाली साइट है तो सेकंड जो प्राइमर है उसकी बाइंडिंग साइट होती है. सो बिकॉज ऑफ दिस रीज़न इट इज़ आल्सो कॉल्ड एज इंटरनल और नेस्टेड PCR. So what may be the basic reaction? Again, we will explain it diagrammatically, that what may be the actual situation or outline for nested PCR. So this process can be divided into two steps, here A and B. So in the first step, the first PCR using external primers, that is, by using external set of primers, we can perform amplification. That is primer 1 and primer 2 is binding here. So we can start the amplification. And normally TAC DNA polymerase, it is being used in this process. And it will lead to the synthesis of first PCR product here. So first PCR product, it is being synthesized by using first set of primer. Now the product of first reaction, now it is subjected to second PCR cycle or second PCR reaction in maybe in a separate vial. So second PCR using internal or nested primers. So second set of primer will bind comparatively internal as compared to the external primer. So primer within first PCR product they will be used. So here we are going to use primer 3 and primer 4, and this will result in the synthesis of second PCR product. So this is the basic outline of nested PCR. Now why we perform or use nested PCR? Sometimes the specificity is required. Fidelity in the synthesis of DNA is required that may not be possible by using basic technique. For example, if DNA fragment is long, so under such circumstances, errors may be introduced. So here we use two set of primers. First set of primer will bind external to the target DNA. It will generate the first product, and then first product it is again used by using second set of primer, so that this reaction will increase the specificity and as well as fidelity in the DNA biosynthesis. And this nested PCR technique It has been used for different purposes, like other techniques. It can also be used in the process of diagnosis of different diseases. For example, this nested PCR technique, it has been used more commonly in the diagnosis of causal agent of tuberculosis like the mycobacterium. Although ये causative agent जो है इसको ट्रेडिशनली जो है वो माइक्रोस्कोपिक मेथड से बाय यूजिंग एसिड फास्ट स्टेनिंग इसको आइडेंटिफाई किया जाता है ऑल्दो इट इज़ आल्सो अ गुड टेक्निक बट सम टाइम high specificity or accurate results may be required in diagnosis. So for this purpose, mycobacterium, it is one of the example that it can be identified by using a nested PCR technique. And there may be some other applications as well.